As an oncologist, I take care of children with high-grade tumors in the brain and spinal cord. Those are the patients who don't have good treatment options at the moment. And I feel very passionate about changing that for them. My name is Mariella Philbin. I am a pediatric oncologist and principal investigator at Dana-Farber Cancer Institute. My hope is that all of our science finally gives a fighting chance to these children and young adults with brain tumors. I want them to come to hospital and hear that there are treatments available for them that will extend their life and make them feel better. We have to go all the way from basic science to translational science to then clinical trials to really make an impact on their lives. Patient tumors are really at the core of everything we do in my laboratory. In terms of the research questions we are asking, we often need fresh patient samples because we want to keep them alive in the laboratory. That means within minutes of that sample coming out of the patient, I will need to bring it back to the laboratory. That of course only works in a setting where the surgeons and the oncologists and the scientists and everybody in between are very much aligned in what they want for the patient. I'm very lucky to have a great collaboration set up with the neurosurgeons at Boston Children's Hospital. So we need to know which patients are being operated on which day, at what time exactly the operation starts, at what time exactly the surgeon expects the sample to be ready. One of the chief nurses calls me from the operating room and says, "Mariella, your sample is ready." I put on my running shoes, run to Boston Children's Hospital, and then I go right into this room where the patient is being operated on. So I'm actually there at the moment when the tumor sample is removed from the patient's brain, which is incredible every single time I see it. And then that tumor piece is put into a plastic tube. I put it on ice and then run back to the laboratory very quickly. I think this collaboration is extremely unique because there needs to be a lot of trust between all the different people involved. When we get those samples to the laboratory, we take a small part and very carefully break it up into little pieces first and then into single tumor cells. If you do it carefully enough, you can on the one hand keep them alive for a few days to weeks, and on the other hand you can look at how they behave with each other in context. We need to learn so much more about the biology of these tumors. What drives them, how are they made, and how can we eventually block their growth for the patient benefit. We are just starting to dig into now how the normal brain is deeply influencing tumor behavior. This is a huge opportunity, we think, for designing new targeted treatments. In my lab, there's people from different backgrounds. For the ones who don't have clinical care as part of their job, they sometimes ask me, like, why are we doing this? You know, isn't it hard to wait for such a long time to bring a basic discovery to the patient? So what I do to help them is I take them with me when I'm in patient service. And I think it's incredibly powerful because it connects research and clinical care in a way that is otherwise hard to explain. You have to be there to see it, and once you do, there's no escape, right? You have to fight for these patients. Very happy to see. I'm very proud that in the last year, 2 of our research stories made it all the way into clinical trials, both of them for high-grade gliomas. Dana-Farber Cancer Institute has a multitude of specialties all working together in a way that I've never seen anywhere else. It's a place where physicians, physician scientists, and scientists work very closely together to make a positive impact on our patients. Every day I work side by side with the best chemists, the best cancer biologists, the best doctors in the field. And to me, that's the only way to make progress.